Alrighty guys, let's get into talking about biomolecules and macromolecules in detail. And so by the end of today, we're going to be able to describe the properties of monomers and the type of bonds that connect monomers in biological macromolecules. And we'll also be able to describe structural similarities and differences between DNA and RNA. Alright? And so think of the four macromolecules, biomolecules we talked about in the last video. What are they and what do you think they're used for? Alright, so think of macros, so things you could find in your food. So carbs, why do you think you need carbs? Lipids, why do you think you need lipids? Proteins, why do you think you need proteins? And DNA, you could also find nucleic acid in your food because living things like plants, animals have nucleic acid. So essentially when you're eating that, you're eating nucleic acids as well. All right, and so carbs are sugars, and they're oftentimes used for quick energy and also for structure. Proteins, they're used for structural and motor actions, and enzymes also catalyze reactions, so they speed up reactions. Nucleic acid is the transfer of genetic material, so it's information storage and transfer, and lipids are fats. All right. So let's get into what is a biomolecule. So it's just a smaller organic molecule joined together to form larger molecules. So here you can see that carbohydrates are made up of these rings. Proteins are made up of these amino acids. Nucleic acids have 3 components to them, and they're completely made out of that. And lipids are the only ones that don't have an actual monomer. It doesn't have any repeating structure. What are they made out of? So you can memorize CHO, CHON, and CHOMP, although lipid has phosphorus, and proteins also have sulfur. But that's like just a way to quickly remember that. All right, and so let's talk about carbohydrates. Carbohydrates are commonly referred to as sugars, as we mentioned. They're made up of monomers, so monosaccharides. Many monosaccharides are going to join together to form polymers. An example of a monosaccharides are like simple sugars like glucose. And polymers, so monomers when they join together, they are called polysaccharides. An example of that are complex carbohydrates like starches, cellulose, etc. And so you might be thinking like, hey, glucose and starches don't taste the same. Although if you think about it, like many monosaccharides join together to make polysaccharides. But remember, structure determines function. So because the structure is completely different, then the function is different, right? So this will taste sweeter and this will taste like starchier, right? And so again, the structure are hexamer rings. So in sugars, you're always going to see these rings. And the main function is short-term energy storage, and also structure. So you see carbohydrates in plants, and it's going to provide its rigid structure. And you also see it in exoskeletons of chitin, so in insects and crabs. All right. And so the elemental composition, as we mentioned, is carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. 1 carbon, 2 hydrogen, and 1 oxygen. So for example, in glucose, you see that in a 1 to 2 to 1 ratio. And glucose is a monosaccharide that is broken down during aerobic cellular respiration to make ATP energy. All right, and so what this means is that basically glucose is the most important monosaccharide because basically it's being used during cellular respiration to make ATP. And if you're wondering what ATP is, it's just the energy currency of our cells. And you find a lot of ATP in our mitochondria. That's why you typically hear people call it the powerhouse of your cell. All right, and so we did mention monosaccharides, polysaccharides, and disaccharides. But in everyday life, you see monosaccharides, for example, in glucose, fructose. Glucose, you can find that in blood sugar, cellular fuel. Fructose is fruit sugar. It's very sweet. And you have another monosaccharide called galactose, which is found in milk sugar. Disaccharides examples include sucrose, which is table sugar, which is just basically glucose and fructose combined. Lactose is milk sugar, which is just glucose and galactose combined. And maltose is malt sugar, which is just glucose and glucose combined. Polysaccharides are starches, like for example, plant energy. They're needed for plant energy storage. And glycogen, that's needed for animal energy storage. Another type of polysaccharide, again, is cellulose, which is going to help plant structure, and chitin, which is going to help make that exoskeleton of an insect and a crab. Do you think you build sugars? Do you think that this is dehydration synthesis, or do you think this is hydrolysis? All right. And so because you see water exiting and you also see something being built up, then you know this is dehydration synthesis, right? You're removing water as a byproduct and you're creating something, so you're synthesizing your sugars, all right? And so you have your two monosaccharides. You're going to get your hydroxyl, so your oxygen and your hydrogen from the second, and you're going to remove that water, and then you're going to form a glycosidic bond, and therefore you're going to create a disaccharide. All right. Again, here you can see the water being released, water being released, water being released. So this is dehydration synthesis. All right. So for example, here you have, and you're going to make those glycosidic bonds. You have water as a byproduct. All right. And so let's talk about some carbohydrate structure and function relationships. Again, because the way the structure is formed is going to determine its function. So here you have starch. It's going to create starch. It's going to be in potatoes. Glycogen, which will be found in your liver. Cellulose, which is found in plants. So for example, algae. And then here you have chitin, which is found in ticks. So the exoskeletons of ticks. All right. And so here's a summary of that. Again, you have your rings. That's how you know this is a sugar. And it's used for structural and storage. All right, so let's talk about branch versus linear polysaccharides. So basically, branch means that there's going to be more surface area. It's going to be a faster breakdown. And linear means that there's stacking ability. So there's going to be strength through hydrogen bonding. So let's get into this. All right, so essentially, energy storage in carbohydrates, that's due to branch structures. So more branches means more monomers can be broken off at once, allowing for cellular respiration to happen faster to make more ATP energy. All right. And so then we have structural carbohydrates that we mentioned, and these are due to the linear structure. So they have the ability to stack. And so here you can see them stacking. And so this gives the carbohydrates the ability to provide tough structural support. And so, like for example, in plant cell walls and in chitin shells and crabs. Alrighty, so digesting cellulose. So only a couple animals can digest cellulose. It's based usually on your diet, your food niche. But for example, gorillas can digest cellulose well, so they must supplement with sugar source like fruit. But cows, they can digest cellulose well because they have this bacteria. And so there's no need to supplement sugars. All right, let's talk about lipids. So lipids are commonly referred to as fats or oils, and they the monomers are fatty acids. And so the polymers, so what it creates is lipids. And so this is for long-term energy storage. And this is because of the covalent bonds. And also can provide insulation and protection of your organs. So in seals, you can see a lot of blubber, and it's for insulation. And it also helps protect our organs. So it has long hydrocarbon chains, which means that it has carbon, hydrogen, and oxygen. There's going to be a lot of carbon and hydrogen and very little oxygen. So here you can see for every 1 carbon, you have 2 hydrogen. All right. And so when we talk about lipids, we also want to mention that there's something called phospholipids that are what makes up our cell membrane. And it's called phospholipids because you do see a polar head and a nonpolar tail. What polar head means is that it's hydrophilic, meaning it loves to be around water, And then our non-polar tail is hydrophobic, so it fears water. All right. And so lipids are hydrophobic, they're going to avoid water. And so when we discuss saturated versus unsaturated fats, there are lipids that show up in many foods. So saturated fats, you could think of those as your bad fats. There's no double bonds, they're straight chains, and they stay stacked tightly. So you're going to see these solid in room temperature. And these can build up in your blood vessels. That's why saturated fats are bad for you. And unsaturated fats have double bonds, so they can create kinks, and therefore they cannot stack on top of each other. And so they're liquid at room temperature, and they're less likely to clog your arteries. All right. So let's talk about building triglycerides. So triglycerides contain glycerol and 3 fatty acids. So here you can see the glycerol and the 3 fatty acids. Tri meaning 3, right? And so what happens is that you have your glycerol with your 3 hydroxyl groups. And so the 3 fatty acids with the COOH groups. And so the 3 dehydration synthesis reactions are going to be 3 dehydration synthesis reactions. So you're going to release water as a byproduct, and you're going to create 3 ester linkages. All right. All right, let's get into lipids. All right. Lipids, as we mentioned, have already been mentioning, are for long-term energy storage. It's for protection against heat loss, protection against physical shock, protection against water loss. The chemical messengers, so lipids do consist of hormones. And the major components of membranes are phospholipids. All right. And here is what I mentioned. It's the stacking, and you do need some enzymes. Then we could get into dehydration synthesis that's needed in order to build these fatty acids. All right, and then we talked about saturated versus unsaturated fats. Saturated fats being the bad fats, and unsaturated being the good fats. So they have kinks, so they cannot stack. Therefore, they're going to be oil in room temperature, and these are going to be solid in room temperature. Again, saturated fats do not have double bonds. They're linear, which means they can't stack at room temperature. Unsaturated fats do have double bonds, and the double bonds in the unsaturated fats prevent them from stacking. Alrighty, so phospholipids in water, so they're going to auto-assemble into something called a bilayer. And so this consists of glycerol, again, and 2 fatty acids. And so for phospholipids, instead of for fatty acids, instead of having 3 fatty acids in fats, we have 2. All right. And they're called amphipathic because it has a hydrophilic head and a hydrophobic tail. All right. So it has both of those polar and non-polar regions. All right. The polar regions are going to be the hydrophilic head, and the non-polar is going to be the hydrophobic tail. And so it's going to auto-assemble into this bilayer again because the tail is hydrophobic, so it's going to be facing away from the aqueous solution in our cell. And so this is going to create a barrier. All right. All right, let's move on to cholesterol. All right, so cholesterol is an important cell component. It's in animal cell membranes, and it's a precursor to all of our steroids. So there are high levels in blood and may contribute to cardiovascular disease. And so just a few atoms create different structures. So remember, structure determines function. All right. Okay, and for steroids, they're 4 fused carbon rings. They have different functional groups that create different steroids. Again, structure determines function. So for example, cholesterol, which is a membrane component, and it's a precursor to other steroids. We have sex hormones as well, so testosterone and estrogen. And so from cholesterol to hormones, there's small differences, but they have big effects, right? They look very similar, but the structure is different, and therefore they have different jobs. And so here's a complete summary. You have lipids, which are your fats, phospholipids, and steroids. Fats have 3 fatty acids, phospholipids have 2 fatty acids, and your steroids have this general structure of 4 fused rings, all right, like we mentioned before. The phospholipids create your bilayer and has those hydrophilic heads and the hydrophobic tails. You have your saturated fats and your unsaturated fats as well. All right, let's move on to proteins. So proteins are commonly found in meats and muscles. The monomers, so they're made up of amino acids, and they create something called a polypeptide, right? So this polypeptide will, they have different structures, but we're going to get into that in a different video. So this is primary, secondary, tertiary, and quaternary, so different folds. And so they have very complex with 4 levels, as I mentioned. So it's going to be more on this later. And we have carbon, hydrogen, oxygen, nitrogen, and sulfur, so chons. And so the main function of proteins are to for wounds and tissue repair, to catalyze, so to speed up chemical reaction. Remember, our enzymes do that, which are a type of protein. For cell signaling, so it sends messages across our cells. And so enzymes are specialized proteins that speed up and catalyze reactions. We talked about that a little in the other video. And so some proteins are embedded in the cell membrane and help with transporting materials into and out of the cell. All right. And so preview for our next lesson, we're going to talk about proteins and how the structure results in different functions. All right, let's move on to nucleic acids. We'll get more into proteins in another video. So nucleic acids, you have, it's commonly referred to as your genetic material that's transferred and stored. It's made up of something called nucleotides, which depending if it's RNA or DNA, it's C, G, A, and U for RNA, and C, G, A, and T for DNA. And it's made up of 3 parts, a sugar, a phosphate, and then a nitrogenous base. All right, a base are those nucleotides. And so the 3 parts are linked by covalent bonds. And so the polymers are the nucleic acids. It's made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus, so CHOMP. And here you see that it has directionality. So it's always replicated from a 5' to a 3'. And you have, for DNA, you have that double helix, and it always will go from the opposite way, just like driving a car, right? You drive a car going this way, and then traffic goes the opposite way, all right? And so the main function, like we mentioned, is to store genetic material. There's 2 types, DNA and RNA. We mentioned how it has directionality with the 5' end and the 3' end. DNA, you could find them in eukaryotes as linear or double, and it's stranded in a double helix. And prokaryotes, it's circular, double-stranded in a double helix. It stores your genetic code. The 4 nucleotides, as we mentioned, are A, T, C, and G. We have a sugar, and it's more stable than RNA. You can find this in your eukaryotic cells in the nucleus and your prokaryotic cells floating around in the cytoplasm. RNA, as you can see, is single-stranded. There's 3 types. You have messenger RNA, transfer RNA, ribosomal RNA. And so you're going to use these for protein synthesis, so in order to create proteins. And there's 4 nucleotides. The only difference between DNA and RNA is instead of having a thymine, it has a uracil. Nucleotides have a ribose sugar in them, meaning it's going to have an OH group, which is called a hydroxyl group. And nucleotides have a deoxyl ribose, meaning it won't have that hydroxyl group, that OH. It's less stable than DNA, and RNA is made in the nucleus, but it can only be used in the cytoplasm, so it's transported to the cytoplasm. So both of them have a 5 to 3 directionality. So it's like driving a car, right? You drive from the front to the back. All right. And so here's a comparison. So they're made up of carbon, hydrogen, oxygen, nitrogen, and phosphate. It's made up of a phosphate, pentose sugar, and a nitrogenous base. The nitrogenous bases, depending for DNA, is adenine, thymine, cytosine, and guanine. And for RNA, it's adenine, uracil, cytosine, guanine. Again, the sugar, RNA is going to have that OH group, meaning it's made up of ribose, and DNA is not going to have that hydroxyl group, so it's called deoxyribose. DNA is double-stranded, as you can see here, and RNA is have that 5' to 3' directionality. So here you can see it goes from 5' to 3', and because it's mirrored, essentially, the other side is the other way, 5' to 3. And something to note is for the base pairing, adenine and thymine right here, they link by hydrogen bonds, and it's 2 hydrogen bonds. And cytosine and guanine link by 3 hydrogen bonds, which you can see here. All right. And so adenine and guanine are called pure rings, and so they have double rings. So you can see right here they have 2 rings. And cytosine, uracil, and thymine are called pyrimidine, and so they're single rings. So you can see 1 single ring, which makes up the basis. All right, and so we have a question right here. So the figure to the left represents a nucleic acid fragment that's made up of 4 nucleotides linked together in a chain. And so here are your bases. Remember, we just said that some of them are have 1 ring and some of them have 2 rings. So which of the following characteristics of figure 1 best shows that DNA fragment is RNA and not DNA? So is it the directionality, the 5' to 3' orientation? Is it the fact that the identity of each nitrogenous base? Is it the charges on the phosphate groups, or is it the type of bond linking the nucleotides together? So what do you think that correct answer is? All right, and it is B, the identity of each nitrogenous base. So based on the bases, they have different structures. And so here you see a uracil structure. It can't be A because they both have a 5' to 3' orientation. It can't be C because both DNA and RNA have negative phosphate charges. So they are negatively charged. And it's not D because both use phosphodiester bonds to link the nucleotides together. All right, so let's see if it's over here. The phosphodiester bonds is what's linking them together. And so the only difference would be the basis, which is determined by the rings. All right, the energy we get from breaking down molecules is called calories. So if we connect this to our daily life like nutrition, we know that there's calories in our food. They're made up of lipids, carbohydrates, and proteins. And so these are all energy needed, and the energy is only going to be able to be used by us by breaking down these bonds and using it for energy. Lipids have the most energy, so like 9 calories in 1 gram of fat. Carbohydrates and proteins have the same amount of energy, so 4 calories in 1 gram. And nucleic acids are not broken down for energy, so you would see 0 calories for metabolism. So you can see how many calories come from the different biomolecules, so the macromolecules, by looking at the nutrition facts on the packaging. So you can see your nutrition packaging and see how many calories they have, how much energy will be provided. So a little review. So compare and contrast biomolecule functions. So put an X in each box that correctly represents a function of the corresponding biomolecule. So for energy storage, do you think carbohydrates are needed for energy storage? Yes or no? And they are. Lipids, are they used for energy storage? Yes, they are. Proteins, are they used for energy storage? They're not. Nucleic acids, are they used for energy storage? They're not. All right, and so remember that carbohydrates have short-term energy storage in your glycogen and starches. Lipids have long-term energy storage, so they have a lot of those covalent bonds that could be broken down for energy. And remember, it has most of our calories. So it has 9 calories per gram. All right. Do these help catalyze reactions? Do carbohydrates help catalyze reactions, so speed up reactions? No. Lipids? No. Proteins? Yes. Do you remember what it's called? An enzyme, right? Nucleic acids, no. All right. Sorry. Coding for traits. Do carbohydrates help code for traits? No. Lipids? No. Proteins? No. And so the only thing that helps code for traits, the only thing that stores our genetic information is nucleic acids. All right. Does this make a part of the cell membrane? Do carbohydrates make parts of the cell membrane? Yes. So it has recognition molecules. Lipids? Yes. So remember that phospholipids make up the majority of our cell membrane. Proteins? Yes. So you do find some transport proteins, some transport protein channels in your cell membranes that let certain molecules that wouldn't be able to normally pass in. It aids in being able to go in and out of the cell membrane. All right. Are they made up of nucleic acids? Are cell membranes made up of nucleic acids? No. All right. Can carbohydrates be broken down for energy? Yes. Can lipids be broken down for energy? Yes. Can proteins be broken down for energy? Yes. Can nucleic acids be broken down for energy? No. So remember, nucleic acids are only molecules that store genetic information. All right, and here's a quick summary. Carbohydrates, remember, it's made up of starches, cellulose, and glycogen. And so they're all glucose polymers. And there's different structures. So they have varying rings that determine its function. And so there could be straight chains, which are used for structure, or branch chains, which are used for energy. And so the properties are that because there's so many hydrogen bonds in cellulose, it's very, very hard to digest. Remember, cellulose is anything that makes up our plants. So that's why only certain animals can digest cellulose. All right, lipids, there's two main parts, fatty acids and a glycerol molecule. And so lipids use a covalent bond called an ester linkage to hold the fatty acids and glycerol together. All right, so there's a very, very strong bond holding this together. So remember that the whether or not there's carbon double bonds will determine whether it's a saturated and unsaturated fat. Saturated fats have no carbon-carbon double bond, so these are going to be solid at room temperature, and unsaturated fats do have that carbon-carbon double or triple bond that could be broken to add more hydrogen to the fatty acid. So these are going to be oils at room temperature. Proteins are made up of monomers called amino acids. And there's 20 amino acids. We're going to go more into this later, but amino acids are made up of an amine group, so NH2. It has a carboxyl group, which is carbon, two oxygens, and a hydrogen. And there's an alpha carbon in the central, to which all the other functional groups are attached to. And there's going to be an R group. That this is different between all of the 20 amino acids, all right? And so amino acids are bonded together by a covalent bond called a peptide bond. And so the peptide bond is a covalent bond and is strong and hard to break. Nucleic acids consist of a 5-carbon pentyl sugar. So we see our sugar right here. It has a phosphate group right here. And it has a nitrogenous base. Remember, depending on whether it's DNA or RNA, it'll have different bases. In our DNA, we see that adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds. I'm sorry, it's very easy to separate them because of the hydrogen bonds are very easy to break. Remember, individually, hydrogen bonds are very easy to break. They're weaker than covalent and ionic bonds. And this is important. The hydrogen bonds being able to break is important because during DNA replication, we're breaking these bonds. During DNA replication and transcription. All right, that's all for this